Yeah, yeah. Good morning and welcome everyone. Thank you for joining us for today's national webinar on molecular docking and in silico ADME prediction organized by Department of Pharmaceutical Analysis to Vidyaniketan College of Pharmacy. My name is Kirti, Assistant Professor, Department of Pharmaceutical Analysis. Firstly, I would like to thank our beloved chairman, Dr. M. Mohan Babu sir, and our dynamic CEO, Vishnu Mandan sir, for always encouraging us to compete and emerge as the best in the dynamic world. On behalf of Shri Vidyaniketan faculty members, I once again welcome all the dignitaries and the delegates of various institutions and the industries for joining today's event, a national webinar on molecular docking and ADME prediction by Dr. S. Juby Ma'am, Assistant Professor. Assistant Professor, Department of Pharmaceutical Chemistry, JSS College of Pharmacy, UT. Shri Vidyaniketan College of Pharmacy, a temple of learning, innovation, and research, always strive towards nurturing and transforming the students into independent citizens and to enhance their quality education. Shri Vidyaniketan College of Pharmacy took its shape in the year of 2004. It's approved by All India Council of Technical Education and Training, New Delhi, Pharmacy Council of India, and affiliated to JTU Anantapur. Now I request our beloved principal and the convener of the national webinar, Dr. Anna Balaji sir, to address the participants of the national webinar.
Madam, am I audible? Assistant Professor, Department of Pharmaceutical Chemistry, JSS College of JSS College of Pharmacy, UT, for accepting our invitation and agreeing to deliver the topic, today's topic, molecular docking in silico ADMET prediction and overview. This topic is of utmost importance in new drug delivery. When you take a look at the pharmaceutical market, the drug pipelines are running dry and many blockbuster drugs are going off patent. So there is an urgent need to bring in more and more new molecules into the market to keep the market going. 59 new drugs were approved in the US in 2018, but in 2019, 48 drugs were shown green light by the FDA, with cancer and rare disease drugs again dominating the list. Historically, drugs are discovered by identifying the active ingredient from traditional medicine or by serendipitous discovery, just like penicillin was discovered by Sir Ian Fleming in 1928. The path of drug discovery is long, challenging, and an arduous task. The cost of drug development is about 1.3 to 2.6 billion US dollars, with an average timeline of about 10 years. In the drug development process, the first step is to identify the target, which plays a key role in the progress of the disease. Once a link has been established between the target and the disease, the next step is to find out potential drug candidates which can stop or reverse the progress of the disease. This process starts with the discovery of the molecules that show efficacy in simple screening process called HITS. Screening is a process in which normally a large number of compounds from natural products and online databases are examined for biological activity in high throughput assays. This step of in drug discovery process is very crucial and demands maintaining huge molecular libraries and carrying out millions of assays. This is a very big disadvantage for individual researchers like academicians and also for small pharmaceutical companies. In case of larger companies, the disadvantage exists in the form of very high cost. Next, the hits are chemically modified to give improved pharmaceutical and pharmacological properties, and such compounds are later termed as leads. The bottom line is that high throughput screening is costly, time consuming, and not feasible both to the academician as well as to the pharmaceutical companies. To overcome these challenges, molecular docking is an exemplary tool. The hit molecules are found from existing chemical libraries using virtual screening. There are more than 1.5 lakh 3D proteins structures deposited in protein data bank. Virtual screening is perfectly viable and an alternative approach to high throughput screening to screen millions of compounds in a few days. Thus, molecular docking and in silico ADMET prediction plays a very important role and cost-effective role in new drug discovery. I would like to thank Dr. Shanmugam, Head of the Department of Pharmaceutical Analysis, Dr. Purnima, Dr. Vijay Mr. Sudhir, and all the other members who are directly and indirectly involved in organizing this webinar. Very soon, we'll be bringing more webinars with subject experts invited from reputed universities, industries, and research organizations. I know this session is going to be an exciting one, and we are all looking forward for Dr. Juby to start off. Without taking much time, I would like to hand over the session to the coordinator. Thank you.
Yeah, thank you, sir, for your valuable words and enlightening speech. Now I request our Vice Principal, Dr. S. Mohan Lakshmi ma'am, to speak few words.
Thank you. Am I audible?
Yes, ma'am. Yes.
Good morning to all of you. Greetings to all the participants here. On behalf of Sri Vidyaniketan College of Pharmacy, I welcome you all for this webinar on molecular docking. Thank you, Dr. Juby, for choosing to be with us today. Hello. And sharing your knowledge with all the participants here. I congratulate the Department of Pharmaceutical Analysis, the head, Dr. Shanmugam, the faculty members, Dr. Vijay Dr. Purnima, Mr. Rajveer, and Mrs. Kirti for taking all the effort for the past week in terms of registration and technicality and all that. So for getting also an overwhelming response that I was told nearly 1,000 of them have registered and are now viewing this webinar. So I congratulate you all for this. So I sincerely believe that this webinar will definitely immensely benefit the participants today, and they will definitely have a good knowledge on this molecular docking information take home with them. Thank you for giving me this opportunity. Thank you, all of you.
From June 2007, she completed her B.Pharmacy in SB College of Pharmacy in the year 1999 and M.Pharm in KM College of Pharmacy in the year 2002 and PhD in the year 2014 from JSS University, Mysore. She has 18 years of experience in both teaching and research. Her research areas of interest are discovery of lead molecules for cancer therapy through insilico drug design. In recent years, she has actively involved in isolation of fatty acids and semi-synthesis of novel bioactive molecules for cancer therapy. She is also an active member in the institutional quality assurance cell. She was a best outgoing student during her M.Pharmacy. She has published around 44 international papers, 21 national papers in both in Indian and also in international with a cumulative impact factor of 49.83. So she has published books and one book chapter and published 3 Indian patents. She has also received 2 research grants sponsored by DST India. And she is also a principal investigator and carrying out the research grants sponsored by AICTE, and it is also sponsored by JSS University, Mysore. She has also received travel grants from DST and ICMR India. She has attended various national and international conferences. She has been invited as a resource person for 2 international and 12 national conferences. She has guided many students, and currently she is guiding 4 PhD students, and she has completed around 15 M.Pharmacy projects. Her biography has been included in famous Nations Who is Who. She is a recipient of Young Researcher 2019 from Association of Pharmacy Professionals India. So with this illustrious career and brief profile about Dr. Juby Jawar, now I call upon Dr. Juby to take over the session. Ma'am, the session is yours.
So thank you, sir, for your nice introduction. So first of all, at the outset, I thank the management, the principal, Head of the Department of Pharmaceutical Analysis, Vidyaniketan College of Pharmacy, for giving me an opportunity to share my experiences on molecular docking and in silico ADMET prediction and overview. Yeah. So in this presentation, I am going to cover an overview of computer-aided drug design. Then I'll be extensively talking on the basics of molecular docking, especially mechanisms, what are the theory involved, what are all the types of molecular docking, and the methodology or procedure involved in the molecular docking. Then I will be moving on to in silico ADMET predictions and ending with a case study which we have incorporated molecular docking and in silico prediction. Yeah. So the majority of the drugs on the market today for treating disorders in humans, animals, and plants were discovered either by chance.
What?
The therapeutic effect. Okay, the people they used to synthesize the compounds, or they used to isolate the compounds from the natural source. Okay, so that is the traditional drug design. Okay, the best example is serendipity drug discovery or penicillin's discovery by Alexander Fleming and all. Okay, so first we generate the lead, then biological testing. If it is promising, then it goes for animal studies. Again, if it is any problematic, then we go for synthesizing or we go for isolating the compounds. So this is the traditional drug design. Now, the traditional drug design, it is replaced with rational drug design. So what is rational drug design? Here, you will have to have a complete knowledge about the molecular targets. Okay, the target is nothing but a biomacromolecule which is involved in the particular disease. Okay, it may be a protein or it may be anything. It is a macromolecule. It is involved in the particular disease. Okay, so this rational drug design has replaced majorly the traditional drug design. Okay, so here the computer technology and the physical principles mainly underlying the drug target interaction. So you should have a complete knowledge about the target. Okay, so in 1980s, the use of computers was extended from data handling to a more prominent role in drug discovery. Okay, so like after this computer-aided drug design has been existed in the field, the use of computers in the field of pharmaceutical research is typically designated as computer-aided drug design or computer-assisted molecular design. So as I told you, so this CAD, it is a combination of so many disciplines. Okay, so you should have a complete knowledge about the physics, because majority of the physical principles underlying the drug target interactions, you will be predicting the binding energy levels. Okay, or X-ray crystallography plays a major role, whether like during the determination of the structure. Then you should have a complete knowledge about chemistry. Okay, then of course biology. You should have a pathophysiology of the particular disease, and you should have a complete knowledge about the molecular pathway of the particular disease. Then only you can able to find out a particular target. Okay, what is of your interest. Okay, so the biology plays a major role, and of course information technology. Without that you can't play a very major role, or you can't do anything. Then statistics and maths, these two disciplines will come for ligand based drug discovery, especially the QSAR techniques. Okay, so you should have a complete knowledge about the statistics and maths also. So CAD is a specialized discipline that uses computational methods to simulate drug receptor interactions to determine if a given molecule will bind to a target, and if so, what its affinity would be. So all these are simulation process only. Okay, how the drug receptor interactions will be happening inside our body. Okay, if it is happening, how extend the affinity, how strongly the target is binding to the particular small molecule. Okay, or so it is all the simulation techniques. Okay, so in the broadest sense, you can call the CAD is the science and art of finding molecules of potential therapeutic value that satisfy a whole range of quantitative criteria, such as example, high potency, high specificity, minimal toxic effects, and good bioavailability. Okay, so this is the process. Okay, and I will tell you very clear. So these are all the CAD tools are mainly used for the prediction. Okay, before touching to your test tube wet lab, no, you can predict and you can start your work with an assumption or with an idea. Okay, so this compound I can synthesize this compound, it will give the activity. So all these predictions and assumptions you can carry out for your further study. Okay, so this is the process. First, you have to identify the disease protein. Then after identification, you have to determine the structure of protein. So here, X-ray crystallography, NMR techniques and all, they play a major role, okay, to give you a three-dimensional structure of the particular protein. Okay, then out of that, you have to identify the active site. Okay, so we know very clear it is a macro molecule. Okay, so you have to find the active site, okay, or the binding packet or catalytic packet. So here effectively the small molecule, it will go and bind. Okay, so after that you can go with visual screening of compound libraries. Okay, now this VS, visual screening, also it is a very emerging technology. Okay, you can divide into two categories: structure based visual screening and ligand based visual screening. Okay, so before, like some 30-40 years before, we are completely depending upon the high throughput screening. Okay, so now this high throughput screening is replaced with this visual screening. So here you will be screening a large number of compounds. We call it as library. Okay, it contains so many thousands or millions of compounds. So those particular compounds you dock or you do the modeling, okay, against a particular receptor or a particular target, that we call it as visual screening. So by doing those, no, you can pick up some potential candidates. So those candidates only you can go for the synthesis. Okay, then after that you go for the pharmacological testing, then you know very clear optimization, then clinical trials as well as the drug. Okay, so what are all the advantages of CAD? So main thing is you can like cost savings. Okay, then time to market. The predictive power of CAD facilitates selection of promising lead candidates, thereby preventing time from being wasted on dead-end compounds. Okay, always we classify two types of compounds. One is hits, other one is dead-end compounds, duds. Okay, the hits, no, you can carry over for your next step. Okay, where this duds, no, it is not that it won't be wasted. It will be like throwing in the dustbin or not. Okay, you can modify it. You can modify into the suitable like lead optimization. You can modify, then you can go for the further step. So the predictive power, it is mainly used for the selection of potential promising lead candidates. Okay, then very importantly, you can get some drug receptor interaction. I told you already, it is all particularly, it is completely based on the simulation, how the target and the small molecule ligand will the action, the drug receptor interactions, it will be taken inside our body. So these are all the very important advantages of CAD. So this is some success story of the drugs discovered through this computer-aided drug design. So it started from 1981, captopril, antihypertensive, and you can see most of the antiretroviral drugs starting with indinavir, ritonavir, saculavir. So these and all, no, they are exclusively discovered through this technology. And this ritonavir has FDA, no, it has a record within 72 days of application, it has given the approval for this ritonavir. Okay, and darsolamide also, carbonic anhydrase inhibitor. Okay, like that, so many drugs you can see here, zanamivir. Okay, so these are all the drugs discovered through this technique. So it is broadly divided into two categories: structure-based design and ligand-based design. Okay, so the structure-based design is nothing but you have a complete knowledge about the target of your interest. Okay, so you have a complete physics and you know complete history of the target. So based on the target, you will be designing some molecules. That is called a structure-based design. The ligand-based designers, you will be having a database of ligands. Okay, where this pharmacophore modeling like 2D QSAR, all 3D QSAR and all, it will come here only. Okay, you have a data set which has all the details of the ligands. From that you design some compounds which will be effective against some particular target. So that is called as ligand based design. Okay, so this is if the structure of the target protein, if it is available, you can go for ligand docking. Okay, information of target protein structures, you can go very well with ligand docking or de novo design. It is a starting with scratch. Okay, you can be completely based on the receptor. Then you can go for molecular dynamics as well as pharmacophore modeling also. Okay, if the target structure is not available, okay, of course you can design the target structure by homology modeling also. Okay, or you can go very well with this ligand based drug design. Okay, where you have the information of the structure of ligands. Okay, so QSAR, you can go pharmacophore modeling and ligand based visual screening. Okay, then you can go for you can get the new lead or optimize the existing lead. You will be getting the drug candidate. So this is different approaches of CAD. Okay, if the receptor is known and the ligand is known, you can go ahead with structure based drug design. If the receptor is known, ligand is unknown, then you can go for de novo based drug design. All the fragment based drug design all comes under this de novo based drug design. Like the receptor is unknown and the ligand is known, you can go ahead with ligand based drug design. Okay, so coming to the structure based drug design, it is called as direct drug design. So when it started in the early 1970s, when the scientists, they had the complete knowledge about the three-dimensional structure of the biomacromolecules. Okay, the revolution of molecular biology started in the early 1970s has led to many three-dimensional structures of biomacromolecules and their complexes with ligands and drugs. Okay, so the ligands can be docked to explore active sites, and de novo design is employed for lead generation and lead improvement. So now coming to the molecular docking. Okay, what it is? So it is an operation in which one molecule is brought into the vicinity of another. So you can calculate the interaction energies of the many mutual orientations and conformations of the two interacting species. Okay, so it is not like that just two molecules will be reacting. Okay, it is a process by two molecules will be reacting. Here the one molecule will be the target, the second molecule will be the ligand. Okay, always it is not that the ligands will be the small molecule. Sometimes it may be proteins also. Okay, protein docking also can happen. What actually happening? So the particular program, it compute the many mutual orientations and conformations of the two interacting species. Okay, so in broader sense, docking is the computational determination of binding affinity between a protein structure and the ligand. So it involves proficient sampling of all possible poses of the ligand in binding packet of the target protein to ease optimal binding geometry as measured by scoring function. Very simple. I'll tell you in layman language. So here, two things you should concentrate. First thing is the sampling or search algorithm. Okay, what are all the different conformations? Okay, both the target as well as the ligand can exist. That is the first part of the theory. The second part is it will give you some output. Actually, it will give some number. Okay, that the number we call it a score. Okay, how the number is coming? So the number is coming from the input of the proper pose. Okay, the software itself, it selects a proper pose and it will give you, it will generate a number as a output. I'll explain in detail in my forthcoming slides. Okay. So the process of docking a ligand to a binding site mimics the natural course of the interaction of the ligand and its receptor via the lowest energy pathway. The energy minimization and the lowest energy should be there. Okay. So different mechanisms. The first classical mechanism is lock and key mechanism. So the molecular docking can be thought of as a problem of lock and key, where one is interested in finding the correct relative orientation of the key which will open up the lock. We know very clear all the keys, no, they won't very well fit into all the locks, isn't it? We should have a proper key to open the proper lock. So here the proper key, in the sense you can imagine in our medicinal chemistry aspect, in the proper orientation, proper conformation. Okay, so that if you have the proper conformation or the proper orientation, then only it will open up the key, then only it will go and effectively bind to the particular target. Okay, so here the protein can be thought as the lock and the ligand can be thought of as a key. Okay, so this lock and key model is the earliest and also the simplest model to explain molecular recognition in enzyme actions. It was first proposed by Fischer in 1894. So as I told you, the lock and key model assumes that the enzyme recognizes the substrate through the shape complementarity between the enzyme's active site and the substrate, which are rigid in nature. Okay, but it has got some drawbacks, because both the here in this, according to this model, both your enzyme as well as the target and the ligand both are kept rigid. Okay, so this model is insightful but too simple to reflect the reality. Okay, so here both interfaces are complementarity in shape and negligible conformational changes take place on binding. Both will be in rigid. Okay, the target is also rigid and your ligand is also rigid. Okay, so here there is no no conformation. Sir, am I audible?
Yes, ma'am, you're audible.
Yes, yes.
So which slide I was not like?
No, till this slide everything was fine, madam.
Sir?
Up to this slide everything is fine, madam.
Okay, sir. Okay. So the lock and key model is the earliest and also the simplest model to explain molecular recognition in enzyme actions. Okay. So what is the major drawback of this? Both will be in rigid, both enzyme as well as the receptor as well as the small molecule. Now, this is replaced with induced fit theory. Okay, so in general, we know very clear both the enzyme, that is a protein, and the substrate, that means a small molecule, are dynamic entities. Okay, they are dynamic in nature, not static, isn't it? That we know very clear. So the comparison between the structures of a protein in complexes, hollow structures, and unbound protein structures, apostructure, demonstrates that the protein often undergoes certain conformational changes when forming the complex structure with its binding partner. So based on this, Koshland has introduced this induced fit theory. Okay, what is this theory? That is both the target as well as the ligand. It can exist in different conformations. Okay, so this model suggests that an enzyme, when binding with its substrate, optimizes the interface through physical interactions to form the final complex structure. Okay, so the induced fit model is supported by the fact that many ligands are buried in the protein binding sites, in the protein ligand complex structures, in the protein data bank. Okay, so you can see here, this is your target. So this target, the conformation has been changed. Okay, it is changed. Same thing, the ligand, this ligand conformation is also changed. Okay, that is what is this theory. Both know they adopt themselves. Okay, they change themselves. Okay, to come to a particular conformation. That is called as induced fit theory. Okay, and of course this induced fit theory also has got some drawbacks. Okay, but for example, such as backbone collective motions, domain rearrangements, okay, and disorder to order transition for highly flexible proteins. Okay, so the side chain backbone collective motions and all not clearly explained by this induced fit theory. So they moved into like I think in late 1990s, this theory, conformational selection theory, that is population shift theory, has been introduced. Okay, so here what it tells suggests that the unbound protein receptor fluctuates among multiple conformational states, with their occupancy probabilities being determined by their relative free energies. Okay, so here the relative free energy take a major role. So here, out of all these conformational states, only a subset of these states allow the binding of its partner. So the encounter with this binding partner shift the distribution toward these states to form the final complex structure. Okay, so what is the main difference between the induced fit model and the conformational selection model is whether the hollow structure pre-exists before forming the complex. Okay, so now recent studies suggest that both models, induced fit, conformational selection, may coexist in many cases. So these are all the different software available. I know very well each one of you might have come through any of this docking software during your research study. Okay, so there are so many freely available softwares as well as commercially available softwares are there. Okay, you can see here Autodoc, it's a freely available software. Okay, then Gold, Glide, Flex, okay, and MOE, that is then even CDocker, LibDocker, okay, Discovery. So all these are all the different softwares are available. Now coming to very importantly, theory of docking, because many of us, we know like we will select a target, we will select the ligand, and you will get the score. Based on the score, you will be moving on to the further studies or not. But you have to understand each and every concept, what it is happening, what is exactly. Okay, so the aim of molecular docking is to give a prediction of the ligand receptor complex structure using computation methods. Okay, the first one is search algorithm. It is a sampling. What that means, conformations of the ligands in the active site of the protein. That is, what are all the various different like possible conformations both the ligand and receptor can exist. Okay, and very importantly, it should be able to reproduce the experimental binding mode. Okay, when you do in real scenario, when you do the real binding assay also in vitro level, so it should mimic the same type of binding modes. That is the very first it's a blend of these two, search algorithm and the scoring functions. What is scoring function? Okay, so this sampling by the search algorithm, you will be getting a fine pose. The pose in the sense, the very good likely very good confirmation of the particular ligand as well as the target. Okay, so that pose, no, the algorithm software, it takes as an input and it gives us output as a number. Okay, and very importantly, what is this scoring function will do? Ranking these conformations via, like among all these conformations, highest most among all generated confirmations. Okay, and this scoring function, no, it also delineate the correct poses from incorrect pose. Okay, not only it just takes the pose as an input and it give you as a output as a number. Okay, it removes the incorrect pose and it selects only the proper pose and it will give you a number that will be called a scoring function. So these two are very, very like basic steps or theories of docking. One is search algorithm, other one is scoring function. Okay, so we have different search algorithms, like starting with matching algorithms. Okay, so this matching algorithms, no, matching algorithms, they are completely based on the molecular shape. Okay, molecular shape into an active site of a protein in terms of shape, features and chemical information. Okay, so all these like pharmacophoric features, you know, very clear, no, hydrogen bond acceptor, donor, ring residues. Okay, we have these pharmacophoric features. So based on these, okay, the selection of this, the sampling will be taken place. The second one is incremental method. So this incremental methods, no, mainly used in fragment based drug design only. Okay, so here the fragment, the ligands, they will be like different broken into fragments, and the fragments also one by one you will be adding on to the you will be trying to dock to the active site of the particular ligand. Okay, so here, no, you use like two algorithms, MCS is multiple copy simultaneous research, okay, and LUD algorithm. The third one is stochastic methods. So here what happening, search the conformational spaces by randomly modifying a ligand conformation or population of ligands. Okay, so here you can divide the stochastic methods into two methods. One is Monte Carlo methods, other one is genetic algorithms. The fourth one is molecular dynamics. So it represents both the flexibility of both the ligand and protein more effectively than other algorithms. Okay, so these are all the different four search algorithms. Each and every software, they utilize these algorithms. Then coming to the scoring function, again the scoring function, you have four categories: classical force field based scoring function, empirical scoring function, knowledge based scoring function, and consensus scoring. So the classical force field based scoring functions, it's very simple. It calculates all the bonding and non-bonding interactions. So bonding interactions means, you know, very bond angle, bond length, torsion angle. So these are all the bonded interactions. Non-bonded interactions like electrostatic forces, van der Waals forces. Okay, so these are all will be like calculating the force. First one, classical force field. The second one is empirical scoring function. The empirical scoring function is nothing but it is the binding energy is decomposed into several energy components like hydrogen bond, ionic interactions, hydrophobic effects. Okay, so this is a simple energy terms. The third one is knowledge based scoring function. It's a very important one. So like here, the statistical analysis of ligand protein complexes, crystal structures complexes, all interatomic contact frequencies, all it will be computed, okay, through this knowledge based scoring functions. The last one is consensus scoring. So this is the combination of different scoring to assess the docking conformation. Okay, so the percentage of strong binder among the high scoring ligand, it will give you the percentage of the strong binding among the high scoring ligands. Okay, so like physics based scoring, this typical scoring function sometimes face the problem of affinity prediction, partly because of the limited treatment of solvation effect. Okay, so this solvation effect, no, it will be computed by MMPBSA, molecular mechanics Poisson Boltzmann solvent accessible area, MMGBSA assay. That means it will give you the binding free energy, molecular mechanics generalized bond solvent accessible area. So the free energy of binding was calculated, which is involved in rescoring or lead optimization to improve the accuracy of binding affinity prediction. So these are all some basic theories. Now we'll come to our this one process. What is happening? You keep a compound in the approximate area where binding occurs and evaluate the following. What are all the things you have to consider? Do the molecules bind to each other? That is the first criteria. If yes, how strong is the binding? Clear. How does the molecule, the protein ligand complex look like? Understand the intermolecular reactions. Then you have to quantify qualitatively as well as the quantitative, quantify the extent of binding. So coming to the different docking methodologies, first one is the rigid ligand and the rigid receptor docking. So when the ligand and receptor are both treated as rigid bodies, the search space is very limited. You can only consider 3 translational and 3 rotational degrees of freedom. Okay. So in this case, ligand flexibility could be addressed by using a pre-computed set of ligand conformations. Okay. So here it won't be, both will be rigid. The second one is majority of the algorithms, majority of softwares it use flexible ligand and rigid receptor docking, where the ligand will be flexible and the receptor is still rigid only. It's a common approach, also a trade-off between accuracy and computational time, is treating the ligand as flexible while the receptor is kept rigid during docking. Almost all the docking programs have adopted this methodology. So this is a simple pictorial diagram where the first scenario, rigid ligand as well as the rigid protein. The second one is what? Flexible ligand and rigid receptor, where only the ligands can be flexible. The third one is both flexible ligand as well as the flexible receptor. Okay, so these are all the 3 scenarios. So the docking can be between protein and ligand, it may be protein and protein, or it may be protein and nucleotide also. Then this is a typical docking workflow. If you take any software, you have to these are all the basic steps. You have to select a target, you have to prepare a target according to the what need of the particular software. Same this side, you have a ligand selection, ligand preparation, then you can go for docking and evaluating docking results. So this is protein data bank. You might be knowing it. It is originally compiled at Brookhaven National Laboratory. So it contains X-ray diffraction and NMR-based structural data of macromolecular structures such as proteins, virus. Okay. So this is, you can see here, as on yesterday, 1,65,422 macromolecular structures have been embedded in this protein data bank. Okay, so from this you can select the target for your interest. Okay, yeah. So this is the selection criteria for protein from PDB. Okay, the first criteria, it is very, very important because you will be having like so many queries, or you will be having multiple PDB codes for each enzyme, each protein. Okay, the first one is the resolution of the structure should be minimum as possible to select. Okay, preferably less than 2 angstrom. That is the first criteria. Now, very importantly, you should have a known binding site, or you should have a natural ligand present which is inside the protein. We call it as co-crystal. The protein structure with known binding site and the natural ligand present in the structure, which is very important for docking studies. And normally we should go for apoform, preferably from human source, and complete structure is available or not, that we have to check it. Okay, and very clearly you have to go for whether your target is wild type or mutant or variant type. So accordingly you have to select it. Okay, then you have to see the latest deposition. Okay, latest deposition. No, the researchers might have deposited the latest years. Okay, or time. So that time period, latest structure, you have to select it. Structures developed by the old techniques might have missing side chains. So right 3-dimensional conformation of side chains is critical in small ligand binding, thus ensure their completeness. Okay. Yeah, and this is one of the step when you are doing the preparation of protein, or when you are doing the preparation of ligands. The one important step is energy minimization. So for that energy minimization, you will be using the different types of force fields. Okay, so the force field is a set of equations and parameters, so which when evaluated for a molecular system yields an energy as a function of the atomic coordinates. So this energy expresses the cost of structural deviation from ideal values. Force fields used in molecular mechanics consider the molecular system as a collection of classical masses held together by classical forces. Okay, so amber force you have, force field, charm force field you have. So this charm is very well used in drug discovery studios. Okay, the chemistry of Harvard molecular mechanics. Okay, then amber is nothing but assisted model building with energy refinement. Then OPLSAH is well used in glide. Okay, optimized potential liquid simulation. Like that you have CFF, consistent force field, then UFF, universal force field. So these are the different force fields will be used for the energy minimization techniques. Okay. Yeah, and this is steps involved in molecular docking with reference. This is with reference to the glide software Schrodinger. Okay, the first one is what? Protein preparation. So you have to go for pre-process. So normally, like during this X-ray crystallography, the hydrogen atoms will be missed. So those and all it will be added. If at all any like side chains are missing, any amino acids are missing, so those and all added through this pre-process. Then you have to go for study of enzyme topology, then removal of non-bonded water, then metal binding sites you have to generate it. Then mainly I told no, you have to minimize the energy by OPLS 2005. Same thing, ligands also you have to prepare, then 3D optimized molecules, then you have to generate the possible ionization states at pH 7 plus or minus 2. Then you have to minimize the energy by OPLS. At last you have to dock. Very importantly, evaluate the docking results and how far you are interpreting. That is very, very important. Okay. So this is a typical example. For this right side, you have all these cow-made analogs, what we have designed, and this is a G score. Okay, this glide score. Also, we have used the glide software. So tamoxifen has been kept as a standard example for the scores. Okay, this is the score. It will give you a number. I told you, no, it's a select a particular pose and it will give you a number. Okay, so this is the G score, is nothing but a combination of all these things like electrostatic energy, Coulombic energy, van der Waals energy, then hydrogen bond, lipophilic, metal binding term, and penalty for buried polar groups, penalty for freezing rotatable bonds. Okay, yeah. And it's an example for interaction studies of amino acids present at binding domain. Okay, so always we have to go for these interaction studies also. Okay, which particular amino acid residue has been interacted with your compound, with your ligand. Yeah, and coming to the ADMET studies. Okay, so the drug molecule not only is expected to be efficacious, but to have ADMET profile, which is normally expressed in the therapeutic index. Okay? So the FDA has approved 117 distinct routes of administration for a drug molecule. So among these 117, oral administration is the most popular one. Okay, so once the drug enters the body starts to process the 4 aspects of a drug: absorption, distribution, metabolism, and elimination. Okay, so why ADMET studies? So desirable absorption, ADME, is very, very important for a candidate compound to become a drug. We know very clear, because you can avoid the late stage attrition. Okay, when you are very thorough or when you are very confident about these ADMET properties. So thus in vitro approaches are now widely used to investigate the ADME properties of new chemical entities. So more recently, the computational modeling has been investigated as a tool to optimize selection of the most suitable drug candidates for development. Okay, so the pharmacokinetic process of a drug answers whether a drug is able to get the site of action. The prediction of the ADMET properties plays an important role in the drug design process, because these properties account for the failure of about 60% of all drugs in the clinical phases. So what are the key objectives? Why we should do this in silico modeling? The main thing is at the design stage of new compounds and compound libraries so as to reduce the risk of late stage attrition. Okay, so when you do the visual screening, no, after that visual screening, there is one screening, it is called as drug likeness screening. So in the drug likeness screening, no, you filter desirable druggable compounds only you will be bringing to the next stage. Okay, all the like non-druggable candidates and all you will avoid it. Okay, so it is very important you can design the library before the in vitro work, so you can avoid the risk of late stage attrition to optimize the screening and testing by looking at only the most promising compounds. So I told you drug likeness, it's a complex balance of various molecular properties and structural features which determine whether particular molecule is similar to the known drugs. Okay, you will be completely computing, okay, by the data of the existing drug molecule, existing chemical databases. So these properties mainly hydrophobicity, electronic distribution, hydrogen bonding characteristics, molecular size and flexibility, and of course presence of various pharmacophoric features influence the behavior of molecule in a living organism. And it was very important Lipinski rule of 5 for a druggable character. For drug likeness, okay, the molecular weight should be under 500, the log p should be under 5, the number of hydrogen bond donors should be 5 or less, the number of hydrogen bond acceptor should be 10 or less. So all are multiples of 5, it will be called as Lipinski rule of 5. Okay, so these are all some important ADMET properties. Okay, so CNS activity, then hydrogen bond donor group, hydrogen bond acceptor. So all for these, no, it will have a standard value. It varies like from each every algorithm to own algorithm. Okay, then QP log hedge, it is nothing but predicted IC50 value for blockade of hedge K channels. Then CACO cell permeability, CACO2 cell permeability, you can predict it. Then percent human oral absorption, okay, then predicted octanol water partition coefficient and rule of 5. So these are all some freely available web servers. You can very well compute the pre-ADMET properties. Okay, so we can give a small projects when the students they are in final year, no, for like 8th semester, we can give a data set, or we can then we can ask them to compute the various physicochemical properties, and we can ask them to correlate the particular physicochemical property to the activity, biological activity. So all those things we can do it. And mole inspiration also, it's a very user friendly web tool. You can calculate all the properties, drug likeness properties. And it is very interesting, Swiss ADME. So here when you open this first page, no, you will get what are all the freely available tools for all of the CAD. Like you go for insilico ADMET prediction or docking also, there will be one click to drug option will be there. Okay, like that you have so many options are there for doing all these freely available. You can use it as a freely available software, and it is well user friendly also. And this is one example for insilico ADMET data where we have computed by using quick prop module of the Schrodinger software. Okay, so here you can see SA1, SA2 and all the designed compounds, and these are all the properties. And down you can see the recommended values where the CNS is -2 to +2, the molecular weight is 130 to 725, dipole, then SA, okay, volume. So everything, even this also like QP log P octanol water partition coefficient, polar surface area, okay, human oral absorption, rule of 5 and rule of 3, all these things. Okay, so by this I'll be finishing the basics of the in silico ADMET prediction as well as the molecular docking studies. Okay, then I'll be moving on to one case study where we have used these molecular docking study as well as the in silico ADMET prediction. The bioactive compounds from spirulina platensis as topoisones. Okay, so it is sponsored by DST Serb Government of India. The principal investigator is Dr. S.P. Dhanapal. Like, he's my PhD guide and he's the principal of JSS College of Pharmacy, Ooty, and myself as a co-principal investigator. Okay, so I have given very snapshots or 10 or 12 slides only, the whole 4 years work. No, I have given where we have used these docking as well as the ADMET tools. Okay, so the spirulina platensis, it's the blue-green algae for free-floating filamentous cyanobacteria characterized by cylindrical multicellular trichomes in an open left-hand helix. It's a potential source of polyunsaturated fatty acids and sulfur lipids. Okay, so I'll be doing so many semi-synthetic analogs of these fatty acids, especially this polyunsaturated fatty acids. So our target of our interest is topoisomerases. So these topoisomerases are nuclear enzymes that play essential roles in DNA replication, transcription, chromosome segregation, and recombination. So all cells contain two major forms of topoisomerase, type 1 and type 2. Type 1 makes single-stranded cuts in DNA, and type 2, which cuts and passes double-stranded DNA. So these compounds active against these eukaryotic topoisomerases are clinically useful anti-cancer agents. So the DNA topoisomerase 1 inhibitor, you might be knowing this: camptothecin, irinotecan, and topotecan. These are all well-known topoisomerase 1 inhibitors. Same thing, topoisomerase 2 inhibitors like doxorubicin and etoposide are currently used in topoisomerase 2 inhibitors. Okay, so these are all this camptothecin derivatives are used clinically for the treatment of colorectal, ovarian, and small lung cancers. Now, what is the research problem? Okay, the existing camptothecin is having a lot of problems like resistance, camptothecin resistance. What are all those problems? Inadequate accumulation of the drug, then alteration of topo 1 resulting in decreased formation of protein linked DNA breaks, alterations in the cellular response to the topo 1 CPT interaction, and very importantly, a concomitant rise in the level of topo 2 expression and vice versa, which leads to the failure of clinical therapies. So in this case, we need a dual inhibitor, okay, so which overcome the camptothecin resistance. So this dual target inhibitors would likely retain cytotoxic activity when resistance was acquired due to alteration of only one target. So however, the physical combination of camptothecin with drugs such as amsacrine and doxorubicin, which also interfere with DNA topoisomerases, often cause antagonistic cytotoxic effects rather than synergistic activity. So in this case, what is the need? A single compound which inhibit both topo 1 and topo 2 may represent the advantage of improving anti-topoisomerase activity. Okay, so there are some drugs like tafluposide, batracycline, and other synthetic or natural products. They have both dual topoisomerase inhibitors, but to date none has been used in the clinic. Okay, so based on these findings, our present work is to discover and develop leads which are targeting both topo 1 and topo 2 enzymes. So these two compounds we have selected for the study. Okay, so here this compound, no, I'll tell you. So this is gamma linolenic acid, a polyunsaturated acid, omega-6 fatty acid. So what we did know, it is a continuation of our work. For the past 6-7 years we are doing this. So we have taken this carboxylic acid, that COH group in the polyunsaturated fatty acid. No, so we have modified that COH group into oxadiazole thiol. It is oxadiazole or mercaptooxadiazole. Okay, So this compound we have taken as one of our compound, and the other one is we didn't modify anything. We have taken a sulfur lipid. So these two compounds we didn't get directly. We have gone for a visual screening. So those visual screening and drug likeness properties, all those things we have got into this two compounds. Okay, then we went for this molecular docking. We have used glide integrated Mastro 9.3 version at that time it is. Then target topoisomerases 1 we have selected 2, topoisomerases 2 we selected 2. Okay, then ligands 5 hepta, that is 2 compounds. One is GLA1, one is a semi-synthetic compound with the gamma linolenic acid analog. The other one is sulfa quinovosyl diacyl glycerol. Then the standard is camptothecin and salvicin. Camptothecin is the topo 1 inhibitor, salvicin is a topo 2 inhibitor. Okay, so first we have gone for this docking studies. So SL showed highest XP glide scores of -7.45 than camptothecin. I'll show you the table, then you can easily understand it. Okay, so here you can see here, like this is the standard camptothecin. So compared to camptothecin, both the compounds are showing very good activity, very good score, particularly the sulfur lipid. Dr. Shanmugam, is there any problem?
Yes, no, ma'am, you continue.
Yeah, the sulfur lipid is having -7.45. So same thing, like one more PDB we have taken. So compared to both the compared to the standard, both the compounds are giving very good scores, G scores. Okay, so the coming to topo 2. Okay, the one the PDB ID is 1ZXN. So we have compared our docking scores with the standard salvicin, which is a known inhibitor. Out of these two compounds, the sulfur lipid, it is showing very good activity, and we have checked with the interactions also. The interactions are also very, very good. Okay, then ADMET predictions also we did. They are very perfect all in all aspects, which activity we wanted to do it. So the design two compounds GLA1 and SL have showed more affinity towards the topo 1 and topo 2 enzymes, and the compound, particularly the sulfur lipid, have showed more affinity towards topo 1 enzyme. However, these two compounds may be further investigated for their topo 1 and topo 2 inhibition. I told you very clearly, it is all prediction only. With this, okay, we can't come to any conclusion. We should prove with in vitro activity. Okay, so we have gone for the lab. So these are all the, just one slide I'll be explaining. The spirulina platensis, the dry powder we have taken, and we have extracted the fatty acid methyl ester. Okay, from that we have enriched the fatty acid methyl ester by using urea crystallization method, where you can get two fractions. One fraction you will be getting the monounsaturated fatty acids and saturated, the other fraction will be polyunsaturated fatty acids. Then we have used our flash chromatography. By using this, we have separated the methyl gamma linolenate. Then we went for the synthesis of that. I didn't show that scheme and all. Okay, then we have confirmed the particular compound by using the mass spectra. It is consistent. Then same thing, sulfur lipid. Sulfur lipid also we have, as such, we have isolated from these spirulina platensis by using flash chromatography system based on the thin layer chromatography system, and the percentage yield was found to 6% weight per weight. Okay, this is the mass spectra. After that, we went for the cytotoxicity studies, okay, by using SRB assay. So the first compound, the GLA1, it is the CTC50 values 0.44 micromolar, and the SL is 0.43 micromolar. Here we have compared our results with the standard compound routine, which is a well-known cytotoxic compound, whereas they are very safe into the normal vero cell lines. Then we went to the cell cycle analysis, which phase of the cell cycle the compounds are very active. Okay, so here the apoptotic populations, okay, is not observed after treatment with GLA1 even at high concentration, but a slight cell arrest is observed in G2M phase. Okay, so in coming to the SL, there is an increase in sub G phase population of cells after treating with 200 microgram per ml of drug. This population represents apoptotic. Then finally, we went for this human topoisomerase 1 as well as 2 assay, in vitro assay. So after that, we concluded that both compounds inhibited topoisomerase 1, which resulted in supercoiled form of the plasmid. Okay, so we what I wanted here, we didn't go for the synthesis or isolation. Okay, so here we have used the molecular docking and in silico ADMET tool for the prediction. With the prediction, we have gone for the synthesis as well as the isolation. So this both works, we have filed and the patents have been published in the year of 2018. Okay, and I told you it is sponsored by the government of India. So these are all some papers where we are using the computational tools for our work. As I told you, I am currently working on this ER positive breast cancer, especially this BRCA1 mimetics. Okay. So I acknowledge, first of all, I acknowledge His Holiness Swamiji, management, JSS Mahavidya Pida, and our Honorable Pro Chancellor, Dr. B. Suresh, our Vice Chancellor, Dr. Surendra Singh of JSS Academy of Higher Education and Research. And I thank my principal as well as he is my guide and mentor, Dr. S.P. Dhanapal, and my HOD, Dr. Afzal Azam, all my colleagues, especially Dr. Gomati Sanesh, and all my PG and PhD students. So they are all working on these, we design the compounds, then only we go for the synthesis. Of course, the Schrodinger, Bangalore, and Altum Technologies, Bangalore. So with this, once again, I thank the management principal of Vidyaniketan College of Pharmacy, Tirupati, for giving me an opportunity to share my experiences. So thank you, one and all. Thank you.
Thank you. Thank you so much, ma'am, for sharing your deep knowledge on molecular docking. Now the session is open for questionnaires.
Still they are listening.
The participants
Those who are having questions can raise their hands.
So that we can unmute them.
Shyam, full screen. Full screen. Full screen is not.
This one.
Full screen.
So participants, Rajesh.
Dr. Vijayaraj, I also request you to check YouTube for any questions there, because a lot of viewers are there in YouTube.
Yes, sir. So we request Rajashekhar Varikala to ask the question. Participants can unmute by themselves after raising the hand. One question at a time.
Vijayaraj sir.
Hello.
Dr. Vijayaraj.
Sorry, yes, sir. Go ahead, sir.
You please announce in the Google chat That Q&A session is on.
Yes, yes, sir. Rajesh Agar Varikala can ask the question.
Go ahead, Mr. Rajshekar.
Yes, sir.
Nagesh, Mr. Nagesh can ask the question. Mr. Nagesh, you can unmute by yourself.
Hello. Hello.
Rajesh Agar Varikala. Anybody having any other questions? Participants, you can raise your hand, you can unmute by yourself.
Hello.
Nagesh, sir, go ahead.
Sir, YouTube chat, you just see, sir. I think some questions
Yes,
are coming.
Raise the hand.
Hello. Hello.
Yeah. We can hear you.
My name is Anguraj from Madras Medical College student.
Okay. Yeah.
Your section is very wonderful and very useful to me. Thank you, ma'am.
Okay, bye. It's okay. It's my pleasure.
My doubt is some proteins in protein data bank have does not have the binding does not have the binding site as well as no co-crystallize with any inhibitor. Then how can we docking? Can we go with the blind docking, or how can we find the binding site in like this protein? Such like, for example, COVID-19 spike proteins, some PDB proteins also does not have the inhibitor binding site. Please clarify that, ma'am.
Okay, yeah. Actually, you there are some algorithms you can design the binding site. Okay, you can design the binding site and you can do the docking studies. I think we have. There are some specific tools. Okay, and coming to this COVID-19, I think there are there are two proteins which we are also currently working on. One is 6LU7, which is having the co-crystal of N3, and other one is also 7BQY. So this is also having the N3 binding site, and so many works are going in. I think you can select 6LU7 or 7BQY. Anguraj.
Okay, thank you, ma'am.
Okay.
Excuse me, ma'am. One of the participants from YouTube has asked whether there is any free software available for toxicology study.
That's what sir, I told you, no, pre-ADMET. In that, no, you can predict the toxicology models. And another one is I told, no, Swiss ADME. So both the tools, no, you can use it. Both are user friendly. You can predict the toxic, this one, what toxicities. Insilico, you can do it.
Excuse me, ma'am. One more question.
Yeah, sir, a little bit. I am not able to hear. Please, you raise your voice a little bit.
One more question from participants. Is it audible, ma'am?
Little lower only voice, little more.
One more question from participants.
Okay.
Is there any software for molecular pharmacotherapy?
What is molecular?
Molecular pharmacotherapy. Yeah, ma'am.
Molecular pharmacotherapy. Maybe I am not getting the question.
Dr. Jubi, can I ask a question?
Yeah,
Principal
sir.
here?
Yeah, please, please.
Very nice session, madam. Thank you.
Yeah, it's nice.
Madam, can you tell me what is the role of artificial intelligence in molecular docking? Is it already started, or it is still in the
But
early phases?
yes, sir, but frankly, I'm telling right now, I don't have any idea about that. But I think it has been started, sir. It has been started, and people are doing that also, artificial intelligence. But so far, I'm frankly telling you, I don't have any idea about that.
Okay. Thank you, madam. Thank you.
Ma'am, another doubt, ma'am.
Yeah.
How can we identify the protein ligand interaction is reversible or irreversible? How can we identify by using a docking score, ma'am?
No, that you have to always the dog scores. No, it can be compared with the inhibitor. Okay, standard standard, whether it is a known inhibitor or known activator. Okay, so by that you can compare, but of course you have to do the in vitro studies only to validate whether the compounds are agonistic in nature or antagonistic in nature.
Okay, ma'am. Thank you. What is the ideal range of docking score, ma'am? Is there any ideal range?
It depends. Usually, that's what I told. It will be comparable with the co-crystal as well as the known standard.
Okay, ma'am. Thank you.
Question అడిగిన hand raise చేయమని.
Any more
Madam,
questions?
I have one more question.
Yes, sir, please.
I am a pharmaceutics background person, so this
Okay,
question
sir.
may look silly for you.
It's okay. No, sir, I have one person at my home itself, a pharmaceutics person. So it won't be. Absolutely not.
So, the thing is, suppose we take any of the existing drugs,
Yes, sir.
phenylephrine or any other drug, paracetamol, simple paracetamol, aspirin. Choose a drug from the shelf that is already, there from long time, and again subject it to these systems. What is the chances, madam? Has anybody tried this such thing?
No, sir. Here, no. Like as far as my concern, they go for this lipid drug conjugate, sir. Actually, what they will do, no, especially like one of our student, no, she is doing that lipid drug conjugate. What she has done, no, sir, that phospholipase, she is targeting that particular enzyme. So she has taken some anti-epileptic drug, and like chemical conjugation, like literally like synthesis. So she has conjugated with the lipid moiety to that particular drug. Then we have checked the docking interactions compared to the pure drug alone. So that in that way we have performed. And now there is a different suits or material science suit, especially for you people like pharmaceutics people. No, I think you can use it more for that material sciences thing. And as far as my concern, we can explore more this ADMET properties, sir. ADMET, especially ADME, and you can correlate to your formulation. So that is my concern. Maybe I have a little idea about this.
No, the reason why I asked this question is now with the existing drugs, they are trying to treat COVID-19 and trying to see which all drugs are effective. You know, it is because no drugs are available and everybody is exploring which drug, existing drugs, are they going to act. So that is the reason I asked this question.
Yeah, sir. Even, sir, like we have tried with one co-drug approach, sir. Like 2 drugs we have conjugated.
Correct.
Yeah, that and we have tried with this M protease, main protease. We have done some work.
Acha, combination of drug docking is also possible.
Yeah, sir. Yeah. Definitely, definitely.
That is new to me.
Thank you, sir.
Madam,
Thank you, madam.
Madam, one more doubt, madam.
Ma'am, one more
Yeah,
doubt.
sir.
Yes, sir. You can go ahead, sir. Hello, Jupi, madam.
Yeah, sir. Yeah. I can hear you.
Madam, up to how many combination of drugs can be taken for the docking study, madam?
Combination.
Yes, madam. Combination of drugs.
Yeah, I think it should be fit into that binding packet. It should be fit into that active site. Suppose if you are ligands, no, the size of the ligand, if it is very big size, no, if again it is very like difficult to explore that. So it is all trial and error basis only. So I tried with two combination, 2 drugs combined together, and we have gone for that, and we have achieved.
Madam, does the molecular weight interferes with the molecular docking, madam? Results?
No, one more.
Molecular weight of the particular drug will interfere with the docking scoring.
What molecular properties, Sandhuma?
Molecular weight, madam. Molecular weight.
Yeah. Of course. That's what the Lipinski rule. No, it says no. The molecular weight, it should be like below 500, and every ADMET tool. No, the basic criteria is molecular weight. Yeah, of course, it plays a major role.
Okay, thank you, madam.
There is one more question from audience side. Is it possible to study synergistic and antagonistic studies through in silico method?
That's what you have to go for in vitro studies only.
Just a question. Hello, can you hear me?
Yes, I can hear you.
Ma'am, just a question, ma'am. Ma'am,
Yeah.
which are now only 12, no? That in docking we have selecting the criteria is there, no, ma'am?
Little louder,
Yes,
Vikram.
Yes, ma'am.
Yeah.
Just you know said that selection criteria is there for docking system, no, ma'am. In that docking system, if you will just you said that resolution structure people is less than 2 angstrom.
2
like
angstrom.
Now only SARX is asked some molecular weight. It is differ something usually.
No. He is, I think Dr. Shanmugam is asking about the ligand, the drug molecule, the molecular weight of the drug molecule. So this is protein resolution of the protein, like macromolecular structure. That should be less than, it should be low, preferably less than 2 angstrom.
It is confirmed. If it is, it's more angstrom means what kind of we can face the problems.
The predictions won't be good. But some other case, no, there is no other go. There is only one protein structure is available in the PDB. In that case, no, you need to go for that only. But always we have choices. So we have choices in the sense, then we have to go, if 2, 3, no, you have to suppose 1.7 or 1.9 means you should go whichever is minimum. 1.7 you should choose it.
Okay. Thank you, ma'am.
Yeah.
Hello, ma'am. One more question.
Yeah.
What is the probability or correlation percent of the in silico data, ADME data with the in vitro ADME data?
Sir, in silico, you are asking about general in silico docking with in vitro activity or alone, only
Yeah,
in silico ADMET?
in silico ADME data correlation with the in vitro ADME data. How much percent correlation will be there? What is the probability?
Probability. So as per my concern, it is not the like what is my I experienced it. Because truly speaking, I stop up to that enzyme activity itself. I won't go for again in silico.
Okay.
As per the papers, as per the research is going on, there is about 60 to 70% correlation, sir.
Okay. Thank you.
Ma'am. Ma'am, good afternoon.
Yeah, good afternoon.
Ma'am, it's a wonderful presentation, ma'am. Small doubt.
Yeah, it's very
Yes,
nice after. Yeah.
Ma'am, is it possible to calculate or detect the adverse effects with that in silico studies? Adverse effects? With the
Adverse
drug
effects,
talking.
yeah, you can go for a probability, like toxicities you can predict. Toxicities you can predict, but adverse effects, I adverse effects, I think I didn't come through it. We maximum we go for this ADME as well as toxicity.
Okay. And any antiviral drugs have been tested with this drug docking for coronavirus? Any idea?
That's what Prandabesh actually like 2 works we have done. One is currently going on. So we have for that we selected some plant constituents and we have gone for visual screening. Then we have identified some potent molecules and all. For that and all we have taken remdesivir as a standard. Though it is RNA dependent RNA polymerase inhibitor, we have taken compared our results with remdesivir.
Okay. Okay, ma'am. Thank you, ma'am.
Yeah. Nice to see you after a long time.
Nice to see you, ma'am.
Yeah.
Any more questions,
sir? Shanmugam, sir?
Yes, sir. Yes, sir.
Our Q&A session was between 12:15 to 12:25. It is already 12:35.
Yes, sir.
I think Madam is also have a long session.
Madam,
enough questions we have taken, I think.
Thank you, madam, for your soft and kind support towards the conference, and your lecture was so wonderful, and we got a nice knowledge from your docking scoring, and we hope we will be having a tie-up with you in future, so our students, our management, and our staffs can come back to you if they have any doubts in this molecular docking. So I suggest you could help us in we are having any problem with the molecular docking, etc. So thank you, madam, for the wonderful session that you have rendered to us. Thank you. Kirti, over to Kirti, madam. Yes, madam.
Yeah,
yes, madam.
definitely. I'm ready to support and share my knowledge, whatever I have acquired. So you are always welcome. So once again, I thank the management, the principal, sir, and Dr. Shanmugam for this opportunity. Thank you so much, sir. Thank you.
Thank you, madam. Thank you so
and
much.
Thank you. Yeah. Shall I leave, sir?
1 minute, madam.
1 minute, madam.
1 minute. I just request Dr. B. Purnima ma'am to give a vote of thanks, to propose vote of thanks.
A graceful and warm afternoon to all. Myself, Dr. B. Purnima, working as Assistant Professor, Analysis Department, Sri Vidyaniketan College of Pharmacy. It gives me honor and pleasure to propose vote of thanks on this occasion. Though the webinar has planned in the short duration and was able to register 1,000 participants in and around India. We received registrations from different states like Kerala, Karnataka, Uttar Pradesh, Madhya Pradesh, Gujarat, Maharashtra, Telangana, and other states. We also received registrations from abroad countries like Singapore, Malaysia, Australia, Ethiopia, Dubai, and other countries. I would like to thank our chairman, Dr. M. Mohan Babu Garu, CEO, Mr. Vishnu Manchu Garu, Director cum Advisor, Professor L. Venugopal Reddy Garu, Director, Quality and Development, Dr. I. Sudarshan Garu, Director on Finance Administration, Mr. B. Ravishekar Garu, for providing us the necessary facilities to conduct this national webinar. In this occasion, I would like to express gratitude towards our principal, Dr. Anna Balaji Garu, who was a guiding stone to the successful completion of this event. Thank you, sir. Now, I would like to extend a hearty vote of thanks to our resource person, Dr. S. Jubi ma'am, who delivered a commendable talk on overview on molecular docking and in silico ADMET prediction. She also discussed about the theories and methodologies related to the docking studies and also selection criteria for the protein docking. Thank you, ma'am. We are really enlightened with your knowledge and presence. I also wish to express my gratitude to our Vice Principal, Dr. S. Mohanlakshmi Garu, for her support to conduct this webinar. A special thanks to our team members, Dr. R. Shenmugh, Pharmaceutical Analysis Department, Dr. S. Vijay Ras, Mr. P. Rajvel, Mistress Kirti, Mr. V. Yogeshwar Rao for their unflinching support, coordination, and work rendered for the last 1 week for smooth conduction of this webinar. I sincerely thank all our MPharm PG students for their support towards this webinar. On behalf of Sri Vidyaniketan College of Pharmacy, I extend my hearty vote of thanks to all our honorable delegates who blessed us with their presence and took out valuable time of their busy schedule. Last but not least,